Hello everyone, in this video we'll be discussing about cytoskeletons as a part of our discussion of cell biology. Cytoskeleton means, cyto means cytoplasm, whereas skeleton means a structure providing support. So these are the structures which provide support to the cytoplasm. These are elaborate network of filamentous proteinaceous structures. They are proteinaceous in nature and they are present in the cytoplasm. We have three important groups of cytoskeletons which include microtubules, microfilaments and intermediate filaments. Let us see about their details. Microtubules. Microtubules generally stay as a tubulin dimer which is made up of alpha tubulin and beta tubulin. Only you need to remember this thing. Whereas microfilaments are actin monomers which are arranged in double helix form. So for microfilaments, just remember actin. For microtubules, remember tubulin dimer, alpha tubulin and beta tubulin. And for intermediate filaments, just remember they are made up of protein subunits. Okay, they are present in the cell. You can see microtubules arising from centrosome, microfilaments encircling the perimeter of the cell, and the intermediate filaments present in the cytoplasm providing support. So let's see the function of cytoskeletons. They provide mechanical support, as the term skeleton itself means. They are helpful in motility. We have seen that the microfilaments are made up of actin, and actin and myosin together forms the muscle, and muscles are required for the motility. And now see the third function, that is maintenance of the shape of the cell. We can easily see that the intermediate filaments are arranged in a cell in the form to protect the shape of the cell. So we have three functions: mechanical function, the second one is motility, and the third one is maintenance of the shape of the cell. Now let's see the uses of these microfilaments. When they come together, they form cilia and flagella. Let us see the biology of cilia and flagella. Hair-like outgrowths from the cell membrane, that is, they both are covered by plasma membrane. So these are the outgrowth of plasma membrane itself. They emerge from the centriole like structure which is called as basal bodies. So cilia and flagella, they are an outgrowth from the cell membrane, and both are covered by plasma membrane, and they arise from a structure which is resembling centriole. Centriole is a non-membrane bound organelle which is useful in cell division, and that centriole like organ is called as basal body. So let us see the structure of flagella. Structure of flagella is made up of three important units. The first one is filament. It is the longest part of the flagella. Second one is the hook, and the third one is basal body through which the flagella emerges. Filament is located towards the outside in the environment, whereas hook and basal bodies are a part of the structure of cell itself. Now let's talk about cilia and flagella. Cilia are smaller when compared to flagella. They work like oars of a boat, or we can say chappu. So what they cause movement of the cell. Either they cause the movement of a complete cell, or they cause movement of surrounding fluid. For example, if we see an example of paramecium, which is a slipper-like organism in which we have a gullet. This structure is called as gullet. This is a paramecium, and this gullet is lined by cilia. What happens in the surrounding? If there is a food particle, this cilia starts to move, and this causes this food particle to enter into the gullet so that it could absorb this as a food vacuole inside itself, and this is a source of nutrition for paramecium. So we have seen that cilia are smaller structures which work as oars. They cause either the movement of complete cell. Either the complete cell will move towards the food, or it will cause the movement of the surrounding fluid or the surrounding medium, so as the food material can come closer to itself and the organism can engulf the food material. The second one is flagella. They are longer structures when compared to cilia, and they are responsible for cell movement. Now let's see that bacteria also possess flagella, but prokaryotic flagella is different from eukaryotic flagella in terms of its structure. We have seen that prokaryotic flagella emerges from the cell wall, and here in eukaryotes we have seen that it is emerging from the cell membrane. Now let's see the electron microscopic structure of a flagella. When scientists saw flagella under electron microscope, they saw this image. Now the main thing was to find out what this actually represents. So after years of research on this topic, ultimately scientists were able to figure out the meaning of each structure present in this diagram, and this was later labeled as per NCERT, which is mentioned in NCERT also. We'll have to discuss this complete structure of a flagella in the following text. So let's start our discussion. First of all, there was a thing called exoneme. Exoneme is a core of cilia and flagella, core pillar of cilia and flagella over which the structure is made up is called exoneme. It possesses number of microtubules. Microtubules are made up of tubulin dimers, that is alpha tubulin and beta tubulin. So this microtubules form the exoneme, which is the core of cilia and flagella. And these microtubules run parallel to the long axis. Actually, what is the image showing is showing the cut section of a flagellum in which the microtubules are running parallel to each other. Now let's see the microtubule arrangement. The microtubules are arranged in two manners. One is the arrangement which is towards the periphery, and one is in the center. So therefore they are peripherally arranged and centrally arranged, in which in periphery they are arranged in 9 dimers, that is radial arrangement, and a single pair, that is 2 microtubule present in the center, which are connected to each other by bridges and are enclosed by a central sheath. You need to remember that microtubules form the exoneme, which is the core of cilia and flagella, and these microtubules run parallel to each other. They are arranged in 2 manners. One is towards the periphery, which are 9 dimers, and one pair which is in the center, which is enclosed by a central sheath and which is connected to each other by a bridge. Only this thing you need to remember till now. And now let's see what is central sheath. Central sheath, which is covering the centrally placed tubulin dimer. It connects to the peripheral doublet. It connects to one of the tubule of peripheral doublet. The central sheath is connected to one of the tubule of the peripheral doublet. A peripheral doublet contains 2 microtubules, and out of those 2, each is connected to the central sheath via radial spoke. So therefore, how many radial spokes would be present? There are 9 radial spokes. Let me explain you with this diagram. So till now we have studied that there is a central sheath which is called as exoneme. So this is a diagram showing a filament. Exoneme is made up of microtubules. Microtubules are arranged in peripheral doublets, which are 9, and a central microtubule, which is a single pair. This is connected to each other through bridge. This is a bridge connecting the central microtubules. Now each peripheral doublet, so this is one single microtubule doublet. Similarly, we have 9 doublets. Okay, I'm not encircling all the 9 pairs. So we have 9 pair of peripheral doublets, and out of these 9 doublets, one of the microtubule is connected to this red color thing. This red color thing is called central sheath through radial spokes. Radial spokes connect one tubule of the peripheral doublet to the central sheath. Understood this much? See, you can easily see that every dimer is connected through radial spoke to the central sheath. I am marking that radial spoke. Therefore 9 radial spokes are present in a filament of flagella. Now let's see what are linkers. Linkers connect peripheral doublet to each other. Therefore this interdoublet bridge can also be called as linker because it is connecting the peripheral doublet to each other. Therefore how many linkers would be present? Of course 9 linkers, because there are 9 peripheral doublets. So there are 9 sets of linkers. Such arrangement of axonal microtubules is called as 9+2 array. That means 9 peripheral doublets and 2 single microtubules present in the center. This arrangement of flagella is called as 9+2 arrangement. That much is clear. So now let's move towards the centrosome and centrioles. Centrosome is an organelle. Centrosome is an organelle which is made up of 2 cylindrical structures which are called as centrioles. We know that centrioles are non-membrane bound organelles. When 2 centrioles come together, they form centrosome. Centrosome is again an organelle. So since they are not covered by a membrane, but they are covered or surrounded by a amorphous pericentriolar material. Amorphous means it is not solid, semi-solid, not solid. Pericentriole, peri means surrounding material. Amorphous pericentriolar materials surrounds the centriole. Centrioles are non-membrane bound organelles just like ribosomes. Centrioles are present only in animal cells and in few lower plants also. But the statement is correct if it comes like centrioles are present only in animal cells. That statement is true. And now see, both centrioles in a centrosome lie perpendicular to each other and each has an organization like cart wheel. Now let's see this diagram of a typical animal cell which shows us centrosome. Centrosome is an organelle composed of 2 centrioles. Centrioles are arranged perpendicular to each other. You can see that they are arranged making a 90-degree angle to each other. Here also the angle is 90 degree. This is 90-degree angle to each other, and in each of the centriole there is a cart wheel arrangement. Cart wheel arrangement. You can easily see this is a cart wheel arrangement, just like a wheel of a cart. Now let's see the electron microscopic structure of a centriole. Centriole are made up of 9 evenly spaced peripheral tubulin proteins. Since this is a tubulin protein, so this is made up of again microtubules, that is alpha tubulin and beta tubulin. Each peripheral fibril is a triplet. If we see this diagram, we can see that in a centriole there are tubulin triplets 1 2 3 1 2 3 1 2 3 again 1 2 3. So these are arranged like a cartwheel appearance, and each peripheral fibril is a triplet we have seen right now in the diagram, and adjacent triplets are also linked to each other. It is very prominent that each triplet is also linked to each other. See via this structure. So it is clear by now that each centriole has tubulin triplet which is attached to each other. And now let's see what is hub. Hub is the central part of proximal region of a centriole. The central part is called as hub, which is a proteinaceous structure. And now let's see what are radial spokes. These connect tubules of peripheral triplet with the hub, and it's also a proteinaceous structure. Of course everything is made up of protein. Now let's see the structure of a centriole. It contains of a tubulin triplet. See, we can count, there are 3 microtubules present, and these are 9 in arrangement. These triplets are 9. 1 2 3 4 5 6 7 8 9. These 9 triplets are also connected to each other through this structure, and in the center we have a structure called as hub. and each triplet is connected to hub also through radial spokes. So these spokes are radial spokes and they are 9 in number, since we have 9 triplets arranged in the periphery. So this is a typical structure of a centriole. The only thing you need to remember, the difference between the structure of cilia and flagella and which is compared to centriole, is that in centriole we have only 9 arrangement, and in cilia and flagella we have 9 plus 2 arrangement, and in the centriole we have 9 triplets, whereas in cilia and flagella we have 9 doublets and 2 isolated microtubules in the center. Now let's see the function of centrioles. Centrioles form basal body of cilia and flagella, and also they are useful in cell division. How? Because they form spindle fibers. In cell division we will study that spindle fibers are fibers which connect to the chromosomes and pull the chromosomes towards the corner of each corner of the cell. Therefore, spindle fibers are necessary for equal distribution of the chromosomes into the daughter cells, and these are formed by the centrioles that give rise to spindle apparatus during cell division in animal cells. You need to remember that centrioles are absent in plant cells, but they are present in only lower plants. And with this we have completed the discussion of the cytoskeleton. Hope you like this video.